scientists prefer to shy away from the religious side of it. Now I want to look at an argument from design, and what we see here is a molecular machine, kinesin, walking up a microtubule, and kinesin transports material from the central region of the cell into outer regions of the cell, and the load is attached to the top. There are many of these molecular machines. This is a very simple one compared to many molecular machines in your body right now, and we'll see a little bit more about that shortly. First of all, a paper published just a few years ago arguing that chance and necessity do not explain the origin of life, and the reason that they hold this is that all known metabolism is cybernetic. This is the abstract, this is their concluding sentence in the abstract. And this is their reason. It's cybernetic, that is, it's programmatically and algorithmically organized and controlled. Essentially what they're saying is it's computer code. And computer code requires that for every point in that code there is a decision being made. And whatever is producing this code must be aware of the different options at each switch point and be able to encode this into the genomes of life. This is the basic summary of their argument. Now, that's just meant to pique your interest. It's not, of course, the fact that two people published a paper saying that, doesn't mean that we should all immediately abandon everything we believed. I want to use an example, however. This is an example of a protein, and to the uninitiated that may look like a bit of a rat's nest, but in reality, about 75% of the proteins in life are composed of a sequence of amino acids such that they fold into a stable three-dimensional structure, something like that. And each structure has its own particular capabilities as far as carrying out some sort of a function within biological life. And some of these structures are so precise that even a shift of an angstrom or two will render it non-functional. Something else you need to know is that the number of sequences that will actually code for amino acid sequences, I should just back up a step. Typically in proteins, there are a sequence of amino acids, and there are about 20 different amino acids. There's 23 found in organic life, but 3 of them are pretty rare. 20 that are commonly used. And so imagine the combination to a safe where you have the average protein has 300 amino acids, 300 to 400 amino acids. So imagine trying to break into a safe where there are 300 numbers, and for each number it goes from, let's say, 0 to 19. So you've got 20 options for each number, and there's 300 of them. That's the average protein. And now typically, unlike a safe where there might be only one combination that will open it, for the average protein there might be a very large number of combinations that will give you the same structure. But in comparison to the number of possible combinations, there are virtually almost 0 combinations that will work. And we'll see some examples shortly. So the other thing that you need to be aware of is that these structures are determined by physics, not biology. And so what biology must do in an evolutionary search is search the sequence space of all possible combinations and find the right combination. So it's really a search problem. If you're going to hold to a Darwinian evolutionary view of the origin and diversification of life, it becomes a search problem in sequence space. With that in mind, we need to know what sort of boundary conditions are involved in this search. And here's some fairly generous parameters. And the bottom line is found here, is that at the most, the total number of trials that will have been available for organic life, if it's been going on for 4 billion years, is about 10 to the 41 trials. That's total for all of organic life. Assuming right off at time 0, you've got 10 to the 30 members in your population, which is estimated to be the total number of life forms on the Earth. Maximum number of generations between you and the first life form is 10 to the 14 generations. So all of this evolutionary search to find all these different proteins, and there are thousands of them, must all take place in 10 to the 41 trials. One of the problems, there are a number of problems with this, but one of the problems is that in these proteins, you can sometimes break them down into modules. Well, I'll call them modules. They're commonly known as structural domains. What that is a section of the protein that will achieve its three-dimensional structure independent of the rest of the protein. And so if you get 2 or 3 of these modules together, they can form the larger protein. And Axe has published some work to try and figure out how often these occur in sequence space. And the best case scenario is about 10 to the minus 64th. In other words, when you look at a set of all possible combinations for the average structural domain, only about 10 to the minus 64th, there's a subset that would be about 10 to the minus 64th of the entire set of possible solutions. And so what you're seeing here is that the target that you're looking for in this evolutionary search is very small. But recall that we only have 10 to the 41 trials in 4 billion years. And so right off the bat, we see we're about 20 orders of magnitude short of what we would require to find even one structural domain, and we need thousands. That's the cold, hard reality that an evolutionary search is facing. So when you hear people confidently talking about how this search did this or that, the other thing, most of the time they haven't even stopped to figure out how many trials are available. In my experience, they have not sat down, and they will not be able to tell you how many trials are available to this search. Here's another way of looking at it. If we're looking at it in units of information, the step size between different structural domains, I would call this a domain set, because sometimes, or perhaps often, we're not sure just how often this occurs. The same sequence will code for maybe 2 or even maybe 3 entirely different folds. You're locked into those 2 or 3, however, depending on how the folding goes. So the difference between, typically, just using axis work of 10 to the minus 64th best case scenario, you would need about 213 bits of information to find the next island. If you're going to think of this in other terms, if the top of this island was 1 square meter in area, and that includes all the sequences that will code for that particular structure, the next nearest island is tens of millions of light years away. That gives you an idea of just how difficult this search is, how impressive it is, what sort of challenges an evolutionary search has available to it. I won't talk about Lenski's simulation. If you're familiar with that, you can raise it, but their step size was radically way too short. Does not really reflect reality. There's a way to check this for yourself. I'm actually giving a lecture on intelligent design on Wednesday, where it's really focused on an argument for intelligent design using a method for ID detection, which is entrenched in science, despite what people might say. We already use ID detection in many ways. In fact, I was using it back in the 80s. Actually, no. It was the 70s. I was working for National Defense Research, writing code that monitored underwater sounds and would filter and look for sounds that were made by Soviet submarines and filter out all the sounds made by natural processes. So it was essentially an exercise in intelligent design detection, in separating all the things made by natural processes, all those sounds, and then looking for just the sounds that might betray human presence under the ocean of some sort. That was back in the 70s. It's also heavily entrenched in SETI, for example. Of course, they're looking for signals that will betray an intelligence out there. Forensic science, it's central to forensic science in distinguishing whether an event or an effect is produced by natural causes or by, what's the typical term? I can't remember the legal term that's often used. And it's also used in archaeology, say ground penetrating radar. You have to distinguish now, is that an ancient buried wall there, or is that just a volcanic dike, or what is that? And so there's constantly this ID detection that goes on in science. Now it's involved in biology as well, as some of you may be aware that the Ventnor Institute has built a genome from scratch for M. genitalium, but they've put 5 watermarks built into that genome that have some of the scientists' names in there encoded. And so now it's an issue in biology. We got to look at DNA and say, is this section of the DNA produced by intelligent design, or is it just natural processes? But